Good afternoon, children. We have started with the 6th chapter, that is molecular basis of inheritance, in which we have studied about DNA, that it consists of nitrogenous base, then a deoxyribose, pentose sugar, a phosphate group. Nitrogenous bases are purines and pyrimidines. Then we have studied about nitrogenous base is linked to pentose sugar through N-glycosidic linkage to form nucleoside. And when nucleoside is linked or is attached to the phosphate group at its 5' position, then it forms the nucleotide, and nucleotide is formed by phosphoester linkage. Then two nucleotides link through 3' 5' phosphodiester linkage, and it forms the dinucleotide, and a polynucleotide chain is formed in this way. And a polynucleotide chain has a free sugar at its 5' end and a free phosphate at its 3' end. Then we have studied that DNA is made up of two polynucleotide chains, and backbone is formed by sugar and phosphate. Nitrogen bases project inside. Hydrogen bonds are present between the nitrogen bases, which hold the chain together. And then we have seen that A pairs with T by 2 hydrogen bonds, and G pairs with C with 3 hydrogen bonds, and the two chains, they run anti-parallel to each other. And the two chains are coiled in a right-handed fashion, and the pitch of each helix is 3.4 nanometer, and 10 base pairs are present in each turn. Then we have studied about packaging of DNA helix, that is in eukaryotes and in prokaryotes, how the packaging of DNA takes place inside the nucleus, that is with the help of the histone proteins. We have studied this. Then we have studied about characteristics of genetic material, with which we can say that both DNA and RNA are the genetic material, but proteins are not the genetic material. Then we have studied about RNA. It consists of nitrogenous base, purines and pyrimidines. The pyrimidines present in RNA are cytosine and uracil. In RNA, instead of thymine, uracil is present. Then RNA also consists of a pentose sugar, which is called the ribose, and a phosphate group. Then we have studied about the Griffith's experiment on transformation. We have studied DNA is the genetic material with the help of Hershey and Chase experiment. Then you have learned about the types of RNA, that is messenger RNA, tRNA, rRNA, and you have studied about the structure of tRNA. Okay, now today we will start with the new topic, and that is replication. Children, in addition to the double helical structure of DNA, Watson and Crick also proposed a scheme for DNA replication. According to this model, the two strands of double helix would separate and act as a template for the synthesis of new complementary strands. After the completion of replication, each DNA molecule would have one parental and one newly synthesized strand. This scheme for DNA replication was termed as semi-conservative DNA replication. Can you see in this diagram, children? The double helical structure of DNA is present. Okay, now the two strands of DNA, that is this strand and this strand, they separate from each other, and the new strand is synthesized from 5' to 3' end. Okay, right. So out of the two strands, one act as the both act as the parental strand. Okay, both are complementary strand, and the new complementary strand for each strand of DNA will be synthesized. Okay, and this scheme of DNA replication was termed as the semi-conservative DNA replication. Now, experimental proof for replication of DNA. Matthew Nesselson and Franklin Stoll in the year 1958 conducted an experiment with Escherichia coli to prove that DNA replicates semi-conservatively. What they have done in this experiment is they grew many generations of E. coli in a medium that contained NH4Cl, okay, where N15 NH4, okay, N15 is the heavy isotope of nitrogen as the only source of nitrogen. Okay, so what they have done is they have taken NH4Cl, okay, where N15 is the heavy isotope of nitrogen and it is the only source of nitrogen. The result was that N15 was incorporated into the newly synthesized DNA. By centrifugation in a cesium chloride, that is CsCl, cesium chloride density gradient, this heavy DNA molecule could be distinguished from the normal DNA. The cells were then transferred into a medium containing normal NH4Cl. The samples were separated independently on cesium chloride gradient to measure the densities of DNA. At definite time intervals, as the cells multiplied, samples were taken and the DNA which remained as double-stranded helix was extracted. The DNA obtained from the culture one generation after the transfer from N15 to N14 medium, that is after 20 minutes, because E. coli divides in 20 minutes, had a hybrid or intermediate density. The DNA obtained from the culture after another generation, that is second generation, was composed of equal amounts of hybrid DNA and light DNA, as you can see in the diagram. Okay, so similar experiments on Vicia faba beans were conducted by Taylor and colleagues in 1958 involving use of radioactive thymidine. The results were that the DNA in chromosomes also replicate semi-conservatively. Children, I hope this experiment is clear. Now we will study about enzymes for DNA replication. The process of replication requires a set of catalysts, that is enzymes, like number one is DNA dependent DNA polymerase, it is the main enzyme which uses a DNA template to catalyze the polymerization of deoxynucleotides. The average rate of polymerization is 2000 base pairs per second approximately. In prokaryotes, there are three types of DNA polymerases, that is DNA polymerase 1. I am not writing this completely. So DNA polymerase 1, 2 and 3. Whereas in eukaryotes, 5 different DNA polymerase have been identified, that is DNA polymerases alpha, beta, gamma, delta. These enzymes are highly efficient as they have to catalyze polymerization of large number of nucleotides in a very short time with high degree of accuracy, as any mistake would result in mutations. Therefore, these enzymes also help in removing mismatched nucleotides by a mechanism called proofreading. Now, the second enzyme is the enzyme helicase. It unwinds the DNA strand, that is, separates the two strands from one point for the formation of a replication fork. The third enzyme which is used is the topoisomerase. The unwinding of DNA creates a tension in the DNA strands, which gets released by the enzyme topoisomerase. The fourth enzyme is the enzyme DNA ligase. It facilitates the joining of DNA strands together by catalyzing the formation of phosphodiester bond. It plays a role in repairing single strand breaks in duplex DNA. Okay, so you have understood which are the enzymes that are required for DNA replication and its functions. Now let us move to the next slide. That is process of DNA replication. Replication is an energy expensive process. It requires energy. Deoxyribonucleoside triphosphate, that is DNTPs, serve the dual purpose of acting as a substrate and providing energy. In a long DNA molecule, replication takes place within a small opening of the DNA helix, which is known as the replication fork, as it is shown in this figure. Now, DNA-dependent DNA polymerases catalyze polymerization only in one direction, that is 5' to 3'. This brings additional complications at the replication fork. Due to this, on one strand, that is template with polarity 3' to 5', replication is continuous. Can you see here the replication is continuous? This DNA strand is called the leading strand, while on the 5' to 3' end, replication is discontinuous. Can you see here? The small fragments are produced. It is not produced in a single strand. Okay. So it is discontinuous, that is in small fragments, and these fragments are called the Okazaki fragments, where the continuous, here you can see the continuous replication takes place and a new strand which is formed is the leading strand, whereas here the replication is discontinuous and the fragments which are produced are the Okazaki fragments, and thereby forming the lagging strand. Now this strand which is formed is the lagging strand. These fragments synthesized are later joined by the DNA ligase enzyme. The DNA polymerases cannot initiate replication process on their own. After replication does not also the replication does not initiate randomly anywhere in DNA. It begins at definite region in a Escherichia coli DNA molecule known as the origin of replication, which is also known as ORI, O-R-I, origin of replication, which has recognition site for DNA polymerase and which also provides site for attachment of RNA primer. In the absence of RNA primer, DNA polymerase cannot add nucleotides. Due to the requirement of the origin of replication, if a piece of DNA needs to be propagated during recombination, a vector is required. The vector provides the origin of replication. In eukaryotes, the replication of DNA takes place at S phase of the cell cycle. After DNA replication, if cell division fails to take place, it results in polyploidy, a chromosomal anomaly. Okay, that means there will be two sets of chromosomes present in the nucleus of the cell. Children, is this clear? Now let us move to the next topic, and that is central dogma. Central dogma was proposed by Francis Crick. In the year 1958. According to the central dogma in molecular biology, the flow of genetic information is unidirectional. Okay, the flow of genetic information is unidirectional. That is from DNA to RNA and from RNA to proteins. Okay, clear? So DNA, it replicates and produces more DNA. This DNA undergoes transcription and produces the messenger RNA, and this RNA undergoes translation and produces protein. Okay, so this is what? This is central dogma. But later in 1970, H.M. Tenim reported that the flow of information can be in reverse direction also. That is from RNA to DNA in some viruses. right, from RNA to DNA, and that occurs in some of the viruses, like in HIV virus, human immunodeficiency virus, which is called the reverse transcription. This is called reverse transcription. Okay. So children, that is all for today. I hope you have understood the replication process very well. In our next video, we will be learning about transcription. So please keep on revising, and if you have any doubts, you can ask me. Thank you.